When treating advanced lung adenocarcinoma, standard platinum-based chemotherapy regimes frequently fail. The tumors adapt, resist the toxic influx, and continue to expand. To understand the mechanics of this resistance, researchers turn to genomic profiling, analyzing transcriptomic data from the Cancer Genome Atlas. The data reveals a tight correlation. High expression of the GGT5 gene aligns perfectly with advanced cancer stages and lymphatic metastasis. This chart displays two Kaplan-Meier survival curves, plotting high versus low GGT5 expression. The cohort crossing the expression cutoff value of 19 experiences a steep, continuous drop in both progression-free and overall survival. Gamma-glutamyltransferases are typically known for maintaining basic redox regulation in healthy secretory tissues. Finding this intense correlation in an oncological setting is highly unusual. High GGT5 expression proves to be a fatal prognostic marker for patients undergoing chemotherapy, directly correlating with treatment failure. But a histological analysis of primary tumor tissues presents an immediate paradox. The malignant lung adenocarcinoma cells contain absolutely no GGT5 protein. This immunofluorescence map reveals the true location of the protein. The green alpha SMA mapping, which identifies cancer-associated fibroblasts, perfectly overlaps with the red GGT5 markers. The protein is confined entirely to the surrounding stroma, creating a bright boundary around the empty tumor cores. Fibroblasts are traditionally viewed as passive structural scaffolding that simply hold tissues together. Here, they serve an active role. Lung tumors survive chemotherapy by outsourcing their metabolic defense mechanisms to neighboring fibroblasts, utilizing a GGT5-driven metabolic bridge. Overcoming this clinical bottleneck requires treating the tumor not as an isolated mass of rogue cells, but as a metabolically integrated microenvironment. Testing this hypothesis requires separating the microenvironment's components. Researchers establish a baseline by comparing primary normal lung fibroblasts against the activated cancer-associated fibroblasts, or CAFs, extracted directly from resected tumors. The CAFs express significantly higher levels of GGT5. They then construct an in vitro co-culture system, pairing these primary CAFs with A549 lung adenocarcinoma cell lines. To isolate the variable, they transfect a specific cohort of the CAFs with small interfering RNA, siRNA, specifically designed to target and silence the GGT5 gene. Researchers extract the conditioned media secreted by both the active GGT5 CAFs and the silenced GGT5 CAFs. Applying these distinct liquid environments to the isolated tumor cells allows researchers to measure exactly what the GGT5 secretion contributes to the tumor's survival. This chart shows the baseline foci formation of the A549 tumor cells grown without chemotherapy. The vertical bars compare the normal CAF media against the SI-GGT5 media. Disabling GGT5 in the fibroblasts causes a significant drop in baseline tumor growth. The researchers then apply a stress test, treating the co-cultures with clinical doses of either paclitaxel or cisplatin. Looking at the updated flow cytometry data for these new datasets, the apoptosis rates spike exponentially, specifically in the cohorts treated with the SI-GGT5 media. The tumor cells possess the exact same internal genetics in both test groups. Their ability to survive the chemical assault changes entirely based on the fluid provided by the fibroblasts. The tumor's chemoresistance is an acquired metabolic shield, supplied entirely by the stromal CAFs. Platinum-based chemotherapies like cisplatin kill cells by inducing an overwhelming surge of reactive oxygen species, or ROS. To survive this oxidative stress, cells require immense reserves of the antioxidant glutathione. Tumor cells lack the metabolic capacity to synthesize this volume alone. This molecular schematic illustrates the tumor microenvironment. Between the tumor cell on the right and the CAF on the left, complex extracellular glutathione molecules drift. The membrane-bound GGT5 enzyme on the CAF surface actively grabs this extracellular glutathione and cleaves its gamma-glutamyl moiety. This cleavage releases cysteine and glutamate into the stroma. These raw precursors drift across the cellular gap and are imported through transport channels on the tumor cell membrane. Once inside, the tumor synthesizes fresh intracellular glutathione, neutralizing the incoming ROS. The CAFs operate as an external metabolic refinery. They process raw extracellular antioxidants into usable molecular fuel, allowing the tumor to defeat systemic chemotherapy. Validating this biochemical mechanism requires moving from in vitro cell cultures to an in vivo nude mouse xenograft model. Researchers introduce a pharmacological intervention called GG-stop, a highly specific small molecule inhibitor designed to block Gamma-glutamyltransferase activity. This graph plots time against tumor volume. The control group receives cisplatin alone, and the tumor volume expands rapidly. The experimental cohort receives cisplatin with GG-stop. This severs the CAF supply line, resulting in a flat trajectory demonstrating profound tumor volume suppression. Throughout the trial, the murine model exhibits no significant body weight loss or systemic toxicity from the inhibitor. Inhibiting the CAF-derived GGT5 successfully dismantles the microenvironment's defenses. Without its external supply line, the tumor's vulnerability to standard chemotherapy is restored in vivo. This mechanism of metabolic outsourcing extends beyond lung adenocarcinoma. Recent transcriptomic studies link other isozymes in the gamma-glutamyltransferase family to aggressive phenotypes across solid tumors. This scatter plot maps the mRNA expression of the GGT7 isozyme across distinct molecular subtypes of breast cancer. High fold change expression heavily populates the luminal A, HER2-positive, and triple-negative cohorts. GGT5 and GGT7 possess distinct amino acid sequences and localize in different tissues, yet they share a deep homology in their redox-regulating potential. Outsourcing antioxidant production operates as a pan-cancer survival strategy. Tumors simply utilize different members of the GGT family depending on their tissue of origin. GGT5 acts as the active engine of a symbiotic lethal tumor defense pact. Traditional oncology often focuses exclusively on targeting the malignant cell, completely ignoring the biological ecosystem that feeds and protects it. Evolving our clinical strategy now requires pharmacological interventions that specifically target and sever the tumor microenvironment's vital redox supply lines. Defeating refractory solid tumors requires dismantling the biochemical infrastructure that ensures their survival.